Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Jonas Buge, Senior Product Manager, Pharma Analytics from Thermo Fisher Scientific.
Great. Thank you, Alex, and for the introduction, and thank you all for joining. Just to set the stage for the next 10 to 15 minutes, you will be hearing about existing and future products in our portfolio and how they can support you in your viral vector analytics journey. We're going to take a look at a standard AAV process and the required residual DNA analytics within. And I will show you some customer data, and at the end, we will provide some information on the use of qPCR and dPCR for these analytics. Okay, excellent. So we already have a number of very well-established qPCR solutions that are easy to use, easy to implement, and that will perform in your AAV process. But especially from gene therapy customers, we often get the question, can you also offer those assays as digital PCR solutions? And to help these customers, we decided to provide QC-friendly solutions on both platforms now, qPCR and dPCR. The fragment sizing kit down here is the first of our launches, and it will be followed by HEC quant assays, residual plasmid assays, E. coli quant assays, so the assays that are really important for gene therapy customers. So now let's take a look as to why they are so important. But to have us all on the same page, this here is a very typical AAV workflow, right? Today we focus on the downstream side. And the E1A, as I just talked about, is to be used to assess the performance of the size reduction steps for DNase or benzonase before and after, and then again at the bulk drug substance level to ensure removal of larger fragments larger than 200 base pairs. And then the other assays to quantitate the residual DNA, host cell, and plasmid are typically used throughout that entire downstream process to ensure proper removal, proper performance of the purification process. So that includes our HEC assay, our CAN-R assay typically. So now let's take a look at a typical sample. These sample schematics, something that we see all the time from customers, very similar throughout, right? And you see right after harvest, you have a lot of residual DNA, plasmid DNA, residual host cell DNA in the sample, larger fragments, and then obviously your viral vector with the gene, partial gene, and also some encapsulated DNA. And then throughout the process, all the way down to bulk drug substance, you see actually that most free-floating DNA, residual DNA in the matrix has been removed, and it's also very small. You also see that the encapsulated DNA is almost unchanged, right? We've lost a few capsids, but what's in the capsid's still in the capsids, and that's obviously not surprising, as the purification process cannot remove residual DNA that is encapsulated. And this encapsulation in itself is really the reason why we have designed this E1A assay, as a lot of our gene therapy customers have approached us and said, hey, regulatories are looking very closely at fragment sizing. Do you have a solution that is really easy and also QC friendly to analyze the size and the distribution of this residual host cell DNA? So that's why we launched the qPCR kit around a year ago, and also our digital PCR kit for exactly that application. So a little bit of statements from regulatory guidances here around residual DNA testing. The FDA, first of all, specifically calls out a 200 base pair limit, right? Therefore, expecting a robust validated method to analyze the residual DNA. And then in addition to that is also a concern for oncogenes, right, that are coming from various regulatory agencies here as well. And if we take a look at the The third one from the EMA. EMA specifically indicates to demonstrate the absence of known oncogenes, right? And especially here, encapsulation in viral vector products. And then now this is a specific concern if your viral vector product is derived from a cell line like HEK293 or HEK293T, because of the containing E1A and also SV40 genes that are part of that host cell. So now to address exactly these requirements, let's have a brief look at the assays that we currently have, qPCR based. And you see the specifications for those assays are all top-notch from a linearity standpoint, from a PCR efficiency standpoint. Precision is all below a CV of 10%, and then also from a sensitivity standpoint. So they're really robust assays. We actually only hear customers being very, very satisfied throughout the years with all those assays. We validate all of our assays to really show they can be validated on our qPCR workhorses, the typical ones that we see in QC environments, which is the 7500 fast, and also the QS5. And to really ensure that customers will be successful with our solutions, we also validate them against the most common gene therapy matrices that are out there, with containing the typical inhibitors that we do see here. So let's move on and look at some customer data. This data here, and I want to mention, is for picograms per titer particles, comes from our qPCR HEK-Quant assay, coming from a standard AAV workflow. And you see here throughout the downstream process, residual host cell DNA actually goes down very quickly, as well as the plasmid DNA. But now the question is obviously, what is that worth, right? And if we take a look at the exact same data set, now only focusing on HEK, and we look at it from a picograms per viral genome perspective, we do see now that for a low dose, let's say E to the 9 viral genomes, very quickly we get below the 10 nanograms per therapeutic dose limit. But if we have to administer a higher dose, 13 E to the 13 viral genomes, we see that we actually never make it below the 10 nanograms per therapeutic dose, and that's just host cell DNA, not even including residual plasmid DNA. So certainly a customer here still has to do some work from a purification standpoint, or better understand what's going on. But what's the main point we want to make here is you see a clear need for a super sensitive and also very representative assay. And since we are talking about E1A in the next slide and also digital PCR soon, I want to make a very important mark here. When working with mammalian genes, large genes, having a representative target is absolutely crucial. And I'm saying this because I see it all the time. Just as an example, our qPCR kit, the one that was used to create this data, is targeting a highly abundant gene that is present in all chromosomes, so super representative. On the other hand, we hear fairly frequently, unfortunately, from customers that are using digital PCR platforms, and they have a self-developed assay, that they try to quantitate residual HEK host cell DNA by targeting just a single gene, often even just a single copy, sometimes just a few copies. And we really highly recommend to not fall into that trap, as a single target with a single copy, like E1A, is not sufficient to reliably quantitate total HEK host cell DNA, right? Especially at lower levels. That's when you really see a difference in results. So switching over to fragment sizing, for this application, and just as a reminder, this is to ensure larger than 200 base pair fragments are removed. For this application, E1A is an excellent target because it really kills two birds with one stone, the way we designed our assay. Because it's quantitating it's analyzing the fragment size for 3 different fragments, short, medium, and long, and it also quantitates E1A as being a known oncogene, which is again another regulatory guidance. You see some correlation. Here we were only testing it at the 3 points that I mentioned before. To HEK, you also see that the customer here in this sample still has some medium and some long fragments in their bulk drug substance present, which is certainly an indicator that they have to continue further analyze what's actually in my capsids here, so that once they go in front of regulatory bodies, they really have a good understanding of the risk profile of this type of product. So switching over to our new assay, right? The results you just saw were created with our qPCR kit. They could have also equally been created with our new digital PCR kit. This kit is very similar to our qPCR assays in the form of appearance, right? It's still an all-inclusive kit with highly characterized standard that comes with it. Specifically here, it is a 3-in-1 singleplex assay designed to target the fragment sizes that I just mentioned. And just to add on to the assay design here, and we've talked to a large number of customers before we designed the assay because there are multiple ways. The design is a nested PCR with overlapping targets so that we are able to generate the highest level of accuracy for the fragments, and that's how the assay was designed. You also see here excellent linearity of the different long fragments, the assays for the different long fragments, excellent separation on the digital PCR instrument, which was used to validate this assay, our new Absolute Q instrument, and also equal quantitation of all 3 fragments throughout the process. So while we are on the topic of digital PCR, just a little bit more on our new instrument. You see it over here. It's actually very small when it sits on the bench. But the ambition of this instrument was really to combine the benefits and advantages of digital PCR with the ease of use of qPCR, right? And the result indeed is a very straightforward qPCR-like workflow. Basically, you mix and spin down your sample, you load the plate and seal it with these gaskets. And then you throw it on the instrument does the rest by itself. Around 2 hours afterwards, depending on the protocol, you can come back and analyze your data, right, with the onboard software. So it's very, very user-friendly, very easy to adopt, and our R&D teams that are now using this instrument very much enthusiastic about the instrument and the simplicity of it. So now let's get to the second last page to go through some characteristics of both technologies from a perspective of residual DNA quantitation, right? And to pick which one is the right technology for me that we want to help you with. So from a qPCR standpoint, obviously it's very well adopted with a huge install base. dPCR, on the other hand, for this application, I would still consider it emerging, right? It's not so broadly installed and utilized. But maybe get there. From a qPCR standpoint, we already have a very high sensitivity, especially with excellent assays. Digital PCR, in theory, a higher sensitivity, for instance, if you're targeting the same single region target. But again, that goes back to the conversation from earlier on if you have a single region target versus a highly abundant, very representative target. So you're losing, obviously, sensitivity there. qPCR readout is directly in mass, which obviously helps for residual DNA quantitation, so no conversion is needed. On a digital PCR instrument, depending on the assay, it's a simple mass conversion, a mass conversion from copy number to mass. Sometimes an experiment is required, really depending on how the assay is designed, what's the target of the assay. So on the qPCR, I would say no surprise, standard curves are routinely required, whereas on digital PCR, it's not routinely required. From a path to regulatory approval, qPCR is so well known, right, and well-established path to regulatory approval. I would say on the digital PCR side, there's still some unknowns in this, and our ambition with the assays that we are designing is to really pave that path so that our customers, with our help, have not too many roadblocks when getting their assays towards a regulatory approval. And then the last thing for the last two bullet points here is around sample preparation and extraction. On the qPCR side, it's best practice, right? And also gives you a very constant LOQ throughout the process. And due to inhibitors, right, often DNA extraction is needed if your matrices contain those PCR inhibitors. Digital PCR is less prone to those inhibitors. So that's why it can happen that no extraction is required. Again, here it heavily depends on the sample matrix and also on the product, right? If you have a lot of protein in your product, better extract it, very likely to obtain reliable results. So that brings us home with my last slide. And just to summarize what we've discussed about today, right? We want to save you development time with our assays and comprehensive product solutions, which are, again, all-inclusive kits with qualified standards, reagents, and the instruments that come with it. We want you to be able to implement those solutions with confidence. That's why we design them with the goal in mind that they can be validated per industry expectations, why they have high, that's why the specifications are set very tightly so that eventually they can be used in QC and for lot release testing. We want to enable you to choose your preferred platform in PCR. That's why next to our qPCR solutions, we are launching digital PCR solutions. And for everyone who is interested in digital PCR solutions, please don't hesitate to reach out to us so that we can make sure that we have the right products on our roadmap. And then lastly, and I think that's a very important point that I always make because it's so important for many, many customers, is that we have a worldwide support network of dedicated field application specialists and engineers that can help with the assays, with software, with regulatory services, with trainings, with the setup, IQ, OQ, etc. Something that we really utilize to partner with all of our customers. And with that, I would like to hand it back to Alex for any questions that are in the pipeline.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, I see a huge value in implementing your off-the-shelf complete kits to measure residual DNA rather than develop these in-house. It's going to make our life much easier. Could you share the data or information around the regulatory acceptance for your ResDNA-seq residual DNA kits?
Yes, absolutely. I think it's a great question and obviously of concern for you by something. Let's take our HEC kit, for instance, on the market for a little over 2 years now. We have hundreds of customers all around the globe in different stages of their journey. And it's a little hard to determine how many of them already went through regulatory approval, but we know that some did, and we never get any problems from customers, any feedback where they need a lot of support from us. So we can say, right, based on the trajectory of the demand of the products, they are working very well in customers' hands, and there cannot be a lot of pushback from regulatory bodies based on that history.
All right, and our next question is, how do you convert copies to NG of ResDNA and dPCR?
Yeah, so it depends on the assay, right, and on your target. If it's a single target, typically we recommend to go with a standard approach of what's the size of your genome, and then you get a copy of one, and then you multiply that with the size of the genome. It's kind of like the worst case approach, but a simple one because it's once established for that cell line, right, you can use that conversion factor, mathematical conversion factor. If you're targeting something that has a larger number of fragments, of targets, then there is clearly a need to determine your correction factor experimentally, right? And that way you can base your future conversion from copies to nanograms off of that kind of like correlation. I hope that answered the question enough. If don't, please reach out to me by email and we can discuss it further.
All right, well, it looks like we're running short on time, so if we don't get to your question, we will be passing them along to Jonas, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, can your ResDNA-seq kits be used on other digital PCR instruments? We don't have the new Thermo instrument.
The digital PCR instruments right now are validated, obviously, on the on the absolute Q. We do know from customers that have been using some of our assays also on other platforms successfully. Some assays did not work, right? Especially if they have a highly abundant target, creates challenges with the technology, and that's what obviously makes it difficult to have an excellent assay right now for digital PCR, but that's why we have those on our roadmap also to support our customers with an excellent solution there.
All right. Well, thank you, Jonas.
Excellent. Thank you all for listening in and for the questions. I'll follow up on the ones that I haven't been able to address right away.
All right. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.